Oh, good morning. The last few lectures you've been looking at how DNA behaves at molecular level, so its structure, how it replicates, structure of chromatin and so on. What we're going to do over the next few lectures is look at how DNA behaves at the genetic level. And what I mean by that is, genetics is the study of inheritance, and the central question of inheritance is, why do offspring look quite like their parents but not exactly like their parents? How is information passed from parents to offspring but varied slightly? This is obviously something that people had noticed for thousands of years, but when Charles Darwin published his On the Origin of Species in 1859, he pointed out that the passing of information from parent to offspring with minor variation provided the raw material for natural selection to act upon. And since it became apparent very quickly that Darwin's theory of evolution was essentially correct, that placed a lot more interest on uncovering the mechanisms for how information passed from parents to offspring. So the real stimulus for the study of genetics came from this work of Charles Darwin. A lot of theories were put forward after that publication for how inheritance works, the mechanisms of inheritance. But it wasn't till the work of a monk called Gregor Mendel, who published in I think 1863, that a plausible mechanism was put forward. And Mendel's big advance, big realization, was that when you're looking at why parents are, or why offspring, sorry, like their parents, in general there's too much variation in parents. And you can see this if you look at any family you know, look at the children. There's a lot of variation between the children. And that makes it quite difficult to work out exactly what information is coming from each parent. There's a lot of variation in the parents, there's a lot of variation in the children. It makes it hard to dissect what information is being passed from parents to offspring. To get over this, what Mendel did was use what are called pure breeding plant strains. You use pea plants. And a pure breeding strain is one that has been inbred for several generations. What that means is that the offspring are consistently just like the parents. So there's very little variation between parents and offspring. And this means, what this allowed Mendel to do was know very accurately what kind of information must be coming from each parent. If you cross two different pure breeding strains, you know what information from each parent will be going into the new offspring. So let's take a look now on the screen at how this worked. Mendel took a pure breeding strain of tall pea plants. They grew nice and tall. And he took another pure breeding strain of short pea plants. They were very short. When he crossed those, he found that all of the offspring were tall. And Mendel explained this by, as simply as he could, by saying each offspring must have some information from one parent and some information from another parent. So there are two bits of information in each offspring. These bits of information he called alleles. And the two alleles put together make up what he called the genotype. It's the combined information in that genotype, the exact combination of alleles, that determines the phenotype. And Mendel realized that you could explain his findings, and I've now got another picture on the screen, if you had, or if you assumed that the tall generation consisted of plants with a genotype capital T, which I've put there on the screen. The capital T allele is what is called dominant. If you have that allele, then you express that trait, the tallness trait. The genotype of the other pure breeding strain, the short pure breeding strain, was lowercase t, two t's. And that meant that the only information that could come from the tall parental strain to the offspring was a capital T allele. The only information that could go from the short lowercase t parental strain was a lowercase t allele. That meant that all the offspring had to have one lowercase t allele and one uppercase t allele. And again, you can see that there on the screen. You can see the combinations in something called a Punnett square, so called because it was discovered or adopted about 100 years ago by a chap called Reginald Punnett. So all the offspring have a lowercase t and a uppercase t allele. Now, because the uppercase t allele is dominant over the lowercase t allele, that meant that all of the offspring adopted the uppercase t trait, appearance. We call that a phenotype. So all of the offspring were tall. Mendel then built on this, and he thought, well, how, if each parent is giving one allele to each offspring, what are the rules that govern the allocation of the alleles from parents to offspring? To work out what these rules were, he crossed the offspring between each other. So he had originally started with two pure breeding strains, the two parental strains. He crossed those to give what is called the first filial generation, or F1, and he then crossed members of F1 to give the second filial generation, F2. And what he found is on the screen now. He found that 3/4 of the F2 generation were tall, but 1/4 were short. Now, what that meant, and again you can look at the Punnett square on the screen, what that meant was that the alleles, the capital T and the lowercase t alleles, must be being allocated evenly and randomly to the offspring. And this is summarised in what is called the principle of segregation, which states that when a parent passes genetic information to its offspring, it does so by giving the offspring at random one of the two alleles present in the parent. The next experiment Mendel did was to say, well, that's what happens if I take the alleles that make up the genotype that is responsible for determining one trait. In the example I've just given, that's how tall a pea plant is. The next thing Mendel did was say, well, what happens if I take two traits, two phenotypes? And he actually tested, I think, 7 different phenotypes, plant height, I think pea appearance, pea colour, a bunch of other things. The example we'll now use is the colour of the peas, colour of the seeds that these things have. And pea plants can be either tall or short, and they can also produce either yellow or green peas. And we can have this on the screen now. If I take, or if as Mendel did, if Mendel took a pure breeding strain of pea plants that were tall and produced yellow peas, and crossed those with a pure breeding strain of pea plants that were short and produced green peas, it turned out that all of the offspring were tall and had yellow peas. That's the F1 generation. That fits with the idea that the pure breeding tall yellow parent has two dominant or two pairs of dominant alleles, capital T at the gene locus that is responsible for height, and then a capital Y, we'll call it capital Y, at the gene locus that is responsible for colour. And similarly in the other parental strain, there is a lowercase t allele at the locus responsible for height, and a lowercase y allele at the locus responsible for pea colour. And again, that's on the screen now. So that means that the only alleles, the only gametes that can be produced by the tall yellow parental generation are gametes with a capital T, capital Y, and the only gametes that can be produced by the short green parental generation are gametes with a lowercase t, lowercase y. When you combine those, you see that every offspring will have a capital T, lowercase t at the the gene locus responsible for height. Now, since the capital T is dominant, that means that all the offspring will be tall. And at the gene locus responsible for the P colour, every offspring will have a capital Y and a lowercase y. So that means that, assuming the capital Y is dominant, all the offspring will have the yellow Ps. Mendel then carried out a second generation cross, so he crossed the the F1 generation with other members of the F1 generation, and that's now on the screen. And he found the offspring adopted phenotypes according to a certain ratio. And you can see the ratio there. 9/16 were tall with yellow peas, 3/16 were tall with green peas, 3/16 were short with yellow peas, and 1/16 were short with green peas. What that suggests is that those two gene loci, the height locus and the colour locus, are independent of each other, and they are allocated at random to offspring. And this led, or this can be summarised in what is usually called the principle of independent assortment, which means that when parents allocate their alleles to an offspring or to a gamete, they do so at random, and the alleles for different genes are allocated to a gamete independent of each other. Now, if we go back again to look at the simple monohybrid cross that Mendel carried out, you can see that there are some tall plants that have a capital T genotype, and some tall plants that have a capital T lower t genotype. So the same phenotype but different genotypes. We often want to work out exactly what genotype an organism has, and we can use the patterns that Mendel found out in his monohybrid and dihybrid crosses, so those 3:1 ratios in the F2 generation for a monohybrid and the 9:3:3:1 ratios in the F2 generations for the dihybrids. We can use those patterns to work out exactly what genotype an organism with a particular phenotype has. And this is done using something called a test cross, where an organism with an unknown genotype is crossed with another strain that has a known homozygous recessive genotype. Homozygous simply means that both alleles in the genotype are the same. So you can have homozygous dominant or homozygous recessive. The pattern of offspring that is then produced by crossing an unknown with the homozygous recessive tells us what the genotype of the unknown is. And you can see an example here on your screen. If the genotype is a capital T lowercase t, then we'll get the 3:1 ratio predicted by the Punnett square. Whereas if it's a capital T, they will all be, or all the offspring will have a capital T lowercase t genotype, and therefore all of them will be tall, have a tall phenotype. So by doing test crosses, we can work out what the genotype of unknown organisms is. So that's a brief summary then of Mendelian genetics. It involves traits, phenotypes, that are determined by a genotype at a single gene locus, where the genotype is made up of a number of different alleles, and the alleles can either be dominant or recessive. The alleles of different genes are allocated to gametes independently of each other. Mendelian genetics, or the model given by Mendelian genetics, is very useful because it provides a conceptual way of thinking about how genetic information is transferred from parents to offspring. Mendelian genetics do actually describe how some traits are passed on. So the P characteristics that Mendel actually studied are transmitted in a Mendelian fashion. However, the majority of traits in the majority of organisms are not, or do not follow strict Mendelian rules. So things like hair colour, eye colour, height and so on in humans don't follow strictly Mendelian patterns of inheritance. And the complications to Mendelian genetics will come across in the next few lectures.